Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Maxim Greenberg from the Institute Jacob Monot on the show. Please let me briefly introduce you to our audience. You got your PhD from UCLA in 2012. You then moved on to do a postdoc with Deborah Bourc'his at the Institut Curie, and you started your own group in 2019 at the Institute Jacob Monot in Paris. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
That goes way back to my youth. I was always interested in science, even at elementary school age. I always liked kind of animals and nature and playing with worms and ants and stuff like that. So I think my roots are very, very deep in that regard. Moreover, I would also like to say that I had scientists in the family. I had an uncle and a grandfather who are both molecular biologists, microbiologists. And so kind of the career path of a scientist, for me at least, was kind of seemed like, okay, this is a viable thing to pursue. It wasn't some abstract thing that only you saw in classrooms or in documentaries and stuff. And so my passion kind of persisted throughout. Biology was my favorite subject in high school. I studied biology in university level and then, kind of went from there. And it's just kind of what I wanted. I had a brief, I would say, a flash in the pan moment where I thought, oh, maybe I'll be a doctor. And in the end, I was taking a medical doctor, and I was taking these biology classes, and I was like, no, I'm taking these classes because I like biology, not because I want to cure people. So it's kind of obvious for me. Medicine is noble, but I really followed my passions.
Oh, that's great. And I guess the context helps, right? If you already are in contact with science throughout your younger years, then I guess it helps to then pursuing a career in science.
Oh no, I think so. I have so much respect for first generation scientists, who aren't really exposed to that in their life, and they really kind of transcend a barrier. In their studies to be like, oh, okay, I'm going to do something new for my family that we haven't seen before, among my relatives. But I have to say, for me, it was like, oh, I could be a lawyer or I could be a scientist. It wasn't, it wasn't something that seemed like a big reach, which is great. I was very privileged in that regard.
So when doing my research, I looked at the paper with your name on it, on PubMed, and it stands out that from your PhD to your current work, it seems to be about the same topic, which is DNA methylation. Was this something you planned? Because sometimes people just change topics when they start their PhD or the postdoc, and then maybe also when they start their own lab. But was this something that you planned?
Not at all. Not at all. I think when I was at university level and even in high school, the aspect of biology that really kind of grabbed me the most was probably evolutionary biology. I didn't study it kind of in a rigorous way in the more kind of higher academic master PhD level, but that was always what kind of I remember like learning about Darwin when I was in 14-year-olds, like, wow, like, that's so cool. But then kind of as I drifted towards molecular biology, I spent a couple of years in biotech, and I was like, okay, well, maybe I'll go into immunology or some kind of something that was kind of more related to human health or something like that, because that's what I was exposed to in biotech. But when I started in my PhD at UCLA, there's different systems, across the world and even in America. But I did 3 different rotations, which I forget how long they lasted. Let's say, 10 weeks or something like that. And I did 3 completely different rotations. I did a rotation in metabolism in mice. I did a rotation in toxoplasma parasitology. And then the last rotation I did, no, I shouldn't say that. The last rotation I chose was in DNA methylation. And that was, I don't want to say kind of an arbitrary choice. I got a really good a sense of my who eventually became my PhD advisor, but I didn't know anything about epigenetics. I just kind of just went there because I had a good idea I had a good feeling of the lab, and it seemed like the lab was publishing all these like really important papers and all these big journals. And I was like, okay, well, I'll check this out, and I just got swept into the subject, and I really never looked back after that. No, but yeah, coming into my PhD, I didn't even know what a histone was. I was coming in very naively.
I mean, now you have maybe, I don't know how much you know about the European system and the US system, but how do you think they compare? Because it seems that you might be able to tip your toe into more waters in the US before you really commit to a career path than you can in Europe.
Yeah. No, this is something I actually gripe about a fair amount. So I've been in Europe for a long time. I got here in 2012. I guess we'll get into that. But something that I really appreciated in the U.S. system, which I think is fairly typical from university to university, is you have these umbrella programs where you can do 3 or 4 rotations that go from 6 weeks to 12 weeks or something like that, which is kind of enough time to dip your toe into a subject. And not too much time where you feel like you lost all this time in your career. And then ultimately, usually what happens, with some exceptions, is you find a good match along the way scientifically, and also the lab you choose, because a PhD commitment's a lot of your time in the prime of your life. You don't want to be unhappy. Whereas at least in France, I don't know how it works in comparatively other countries in Europe. I know there are certainly some differences, but yeah, essentially the master's internship you do in France before you do your PhD is very long. It's 6 months, and if you make a bad choice 1 month in, you can really be shooting yourself in the foot, and in the lab too, because they make a big commitment to you as well. So yeah, I think in that way, I mean, there's pluses and minuses to every system, but in that way, no, I think I really prefer the way that the Americans have kind of set it up.
Yeah. So let's start with your PhD work. There, working in Arabidopsis, you identified factors involved in the de novo DNA methylation. Which approach did you use in the study, and what did you find then?
Well, so yeah, this was a really exciting time. So the epigenetics field, as you know, is not that old. And then my PhD advisor, I think he kind of stumbled into epigenetics himself in the late 1990s. I think he was studying floral development, and he ended up finding an allele, a gene that he thought was mutated, but it was actually silenced by DNA methylation. That was really kind of his jumping off point. So that he was doing a lot of genetic screens to find new factors, which was very common. And Arabidopsis is a great model for genetic screens. So the first project I had was a genetic screen. So the DNA methylation establishment pathway in Arabidopsis is actually a small RNA pathway, which was also very exciting, because it was kind of this confluence of small RNAs, which had won the Nobel Prize very recently, and then also DNA methylation epigenetics was all kind of coming together. And so the screen was designed by a postdoc who actually had left the lab prior to my coming in there, and the framework is that he could introduce a transgene that if it's DNA methylated, it's silenced, and if it's not DNA methylated, it's active, and if it's active, then the plants flower late. So it's very easy to score. Okay, so you introduce this transgene, and if there's no de novo methylation, the plants flower late. And then after that, you do your mapping crosses, and you find your genes of interest. And so that was kind of my primary project coming in. And I would say it was exciting. I would say the bad news is that our lab wasn't the only lab doing genetic screens at the time. This was a very hot, I would say white hot period of DNA methylation screens in Arabidopsis. So there were a few other really, really great labs. And so every time we found a factor, it was like, oh, that one was just published in Nature a month ago, or we know these other people are working on it. So we didn't get anything that was, I would say, extremely novel. I did have one kind of amazing period where I did get a novel factor, but it happened that somebody else in the lab doing another approach biochemically also discovered that same factor. So, you had this kind of convergence of events. And it's a great pathway because, there are, I don't know how many total factors there are, but it felt like dozens of factors. So it was just the screens were very rich coming out, so that was really exciting. So I think, coming in, I really love genetics. And I think my PhD advisor, he was kind of more of a trained geneticist, but during that time the lab was also shifting towards some proteomic methods like IP mass spec, which was exciting. And then we weren't doing structural biology ourselves, but we had collaborators who were doing crystal structures of the factors we found. So we were kind of publishing these papers with these I think very well-rounded characterization of these factors. Moreover, we were really kind of leading the way on genomics and epigenomics. So we did, publishing whole genome by sulfite maps of the mutations and stuff like that. So it was great. It was, and it felt like so easy to publish at the time. You just get something new and publish it.
Yeah, coming back to the publishing and that people were working on the same things that you. I mean, at least you knew that you were working on something relevant, right?
Yeah. Well, that we told ourselves. Yeah, I think that it was. Why these things get so exciting? That's I don't know. It's kind of in the ether, but it felt exciting. It felt like it felt important. I mean, the irony is that in the I guess we'll talk about mammals in a little bit, but if you take out the factors that are required for DNA methylation establishment in mammals, you've got these really severe developmental phenotypes. And the strongest being pretty strong embryonic lethality. Whereas if you take out all these factors that are required for DNA methylation in plants, the plants look basically wild type. They look totally fine. So we have these mutations, and we're publishing them in Nature and Science and wherever, and the plants are totally healthy and fine. You're like, so what? what's so important about this? But we can talk about why it is important. But yeah, no, I think it's kind of funny in that regard. Yeah, but yeah, no, it was really exciting.
Yeah, yeah. Now I need to decide whether I want to go down the path you just mentioned, or with my next question. Maybe we do both, just one after another. So the next paper I found was mentioning Jumanji 14. And LDL1 and LDL2. Was the Jumanji one of the factors that you mentioned that you found in those screens that were kind of relevant or important?
No, we didn't get it from a genetic screen. Actually, the DNA methylation phenotypes in those Jumanji proteins, or those mutations in the genes that code for those Jumanji proteins, they were pretty mild at the level of DNA methylation. I think, this is also another time. So, as you might know, I think it was around 2007 or so not so long after I started my PhD. This is when the Jumanjis were first discovered. And so, all of a sudden there was a lot of excitement about those, because I think for years, smart people thought that there were not even factors that could demethylate lysine residues on histones. Just thermodynamic. I'm a biochemist, but thermodynamically, it's something that would be too difficult for an enzyme to overcome. So when it came out, of course, there was a lot of interest in that. And then you could predict the Jumanjis just from their sequence. And of course, the Arabidopsis genome was sequenced. So we had a collaborator, a guy named Daniel Schubert, who's in Germany. I forget his institute at the time. I think some Max Planck in Germany. And he had this collection of Arabidopsis Jumanjis. And he sent the seeds over to my postdoc advisor. And then Angelique Delarice, who was a postdoc in the time, she started characterizing them, and then I kind of jumped on those. And it was kind of fun because, we ended up working with these Jumanjis, and the ones that ended up having the phenotype made sense. They're the ones that were demethylating H3K4. And H3K4 methylation, demethylation, there's this kind of conserved antagonism in plants and animals between them. So it kind of made sense that these could be working to help with establishing demethylation.
So before we move on to your postdoc, which was with Deborah Bourc'his, so those two papers I found were where you were a main contributor, but you mentioned that you had many other things coming out that were published in high-impact journals. So what was there that we didn't mention about your PhD that maybe is worth mentioning?
Yeah, so, we are also, there was a lot of projects kind of humming in the lab. I thought the way that the lab was
just
run.
to make the picture clear, which lab was it?
Yeah. So this was Steve. Oh, I'm sorry. We should have mentioned this. So this is Steve Jacobson's lab. So this is really kind of one of the leaders of DNA methylation research in plants. I would say in general, but certainly in plants. And he's really done a lot of major discoveries, since he started his lab, in the last couple of decades, and continues to. He's really been a leader in the field.
I should ask him to be part of this podcast as well.
Oh, yeah. I mean, yeah. Well, he would. He's got a lot more to talk about than me, I'm sure. I mean, he's just had decades of just phenomenal studies and papers coming from his lab. But so we were, it was a really exciting time, as I mentioned. There was a lot of postdocs in the lab who were really hardworking, really smart, and just had a lot of projects kind of running. And you would kind of, and then there was a really friendly group too. That's something kind of I learned from him, and as well as my postdoc advisor, to kind of keep this really, I would say, jovial atmosphere in the lab. Of course, it was competitive and ambitious, but at the same time, didn't feel like people were stepping on each other's toes, or there was intra-lab competition. It was more synergistic. People were working together, and so there was a Spanish postdoc named Israel Asin who was working on these RNA binding proteins, and then one of which I actually stumbled in from the screen. So we kind of started working together. And we published, a paper on these. They ended up calling them IDN proteins, which where they're placed in the pathway. They seem to be downstream of small RNA biogenesis, but they bind to RNAs. So maybe they're helping link the DNA methylation machinery with the RNA pathway. That was kind of the model at the time. So we published how many papers did we get on that? I think we had one PNAS paper on that where I was a co-author with Israel, and then the two papers on the Jumanjis and the paper on the screen, and then and then there was one last. There's a couple of papers that ended up as not the first author, but kind of a middle author, because either I discovered things from IP mass spec or they came from the screen. So I had data that I shared with people working on things that were going on in parallel. So it was really fun.
So when I look back at my PhD and comparing it with your output, it was quite productive what you did in your PhD, right?
Yeah, well, I don't know. It's funny now, all these years later. Now it feels very hard to publish. I don't know, I guess things have changed. It certainly feels like you just need a lot more data now if you want to get published into a high impact journal. But I had a very informative experience when we published on that Jumanji 14 factor. So that was with Angelique, who I mentioned, a postdoc in the lab. And, we knew that there was competition. And so we're like, okay, well, we have this data. Let's put it together into a paper. And, I think by today's standards, it felt very minimal. We characterized the DNA methylation. We characterized the de novo methylation phenotype, which was late flowering. And, I think maybe we did immunofluorescence and some CHIP, and it was out. And we got a paper in Ember Reports, which is a very good journal, and it's like, wow, this is what you need for a paper. It felt like, by today's standards, very, very minimal. Now you just need so much more. That's another topic, I suppose.
Yeah. So then you moved on to Deborah Bourc'his' lab, which we already mentioned. So I don't know how many Americans come to Europe to do a postdoc, because there are so many great labs in the US as well, right? So making the switch and traversing the Atlantic is maybe something that not so many Americans do. So maybe you can just talk a little bit about how this came into place, and then let's talk about the science later.
Well, so something I would say, I wasn't as much as it wasn't my plan to study epigenetics, it wasn't my plan to come to Europe either. I thought, America's a big country. I don't have to, that's not breaking news, so you can travel big distances in America and still stay in America. And that's kind of typically how Americans work. You do your PhD in Harvard and you do your postdoc in Stanford. And, that's a huge distance. It's like going from Madrid to Moscow, essentially, But on the other hand, when I think there were some personal reasons for me to come to France, because my now wife is French, so we wanted to kind of try something new, and she was very interested in coming back to France. So in that regard, it's like, okay, well, we'll give it a shot. That said, that now that I've done it, it was such a great experience, and I wish more Americans were more open to this, because I think. Really, going from Harvard to Stanford, it's a big physical distance, but culturally, it's very, very similar. Whereas you come to Europe and you see how science is done, coming through the lens of another culture, I think that can really open your mind, and I think make you a stronger scientist. And there's a lot of great labs. I don't have to tell you, in UK and in Germany and in France, and in the Nordic countries, there are these fantastic labs that Americans, I think, would be thrilled to be in. And, there are some, it's a minority. America's a big country. And, I think they kind of fall into three groups, kind of the really courageous ones, the ones who came for personal reasons like me, or the ones, you see them in France, certainly, the ones who studied French also and loved French culture, and, that kind of
all good reasons.
Yeah, for sure. But and I haven't looked back. We've been very happy here, and so far, knock on wood, things have been very smooth staying here.
So in Deborah's lab, you switched to the mammalian system and demonstrated that chromatin changes that occur in the pluripotent embryo can be dispensable for embryogenesis, but instead signal essential regulatory information later in the adult, which is
yes.
Very interesting to think about how this delay can take place. Can you talk about the study and what you found?
Yeah, sure. I mean, this was so, one of the reasons that I was drawn to mammals is because, there's this phenomenon called DNA methylation or epigenetic reprogramming that is very mammal specific. As far as we know, it doesn't even happen in other vertebrates. There's this embryonic wave of demethylation where essentially the gametic demethylation patterns are not completely lost, but, substantially lost. And then as the embryo is, eventually implanted into the uterus, and then you go through all the differentiation, gastrulation. But just prior to that, there's this wave of remethylation where the DNA-methylation levels genome-wide become very highly methylated. And I think what stood out to Deborah and me is that once they're established, and we're talking in the mouse, this is about, 1 week after fertilization. In the humans, about 2 weeks after fertilization. So essentially in a period where a woman wouldn't even know that she's pregnant. The demethylation levels are established, and from that point forward, there is at least the latent potential for those patterns to be maintained throughout life, because demethylation can be very stably maintained in an epigenetic manner. So, the gene that I ended up characterizing, and I can't quit it. I just keep characterizing it because it's a really interesting gene. It was found earlier from a previous project done by Charlotte Proudhon, a former PhD student of Deborah's. What she found was in a specific mutant, when there was higher methylation, they saw higher expression of the gene. So that was pretty interesting. And so I came in to kind of jump in and figure out, okay, what exactly is happening here? And so what we ended up finding is that the DNA methylation that was upstream of this gene, it was actually antagonizing silencing complex called polycomb repressive complex 2. So denamethylation we always think of as repressive, and it certainly is. Transposable elements probably most notably. But in this case, the denamethylation seemed to be protecting this gene from polycomb-mediated silencing, and this allowed for expression. So that was already great, because I love epigenetic mechanism, and I probably at my heart, like, that's what I'm most interested in, is really kind of this mechanistic aspect. But I was working with a really talented PhD student named Julian Glaser, who now is actually about to start her own group her independent group in Max Planck in Freiburg. And maybe one day you'll have her on as a guest as well. She's fantastic. But anyway, so what she did in parallel to my mechanistic work was we made a so-called epi-mutant where there was no DNA methylation at the ZDBF2 gene. And what she observed in the mice was that actually they were smaller. And, from our work in embryonic stem cells, we knew all these mutations were doing, they were totally fine. And when she looked in her embryos, those embryos, as far as they could tell, were totally fine. So this seemed to be this programming of this gene, which seems important in postnatal life, in appetite control. So that was really exciting, and that kind of like was kind of the launch pad for my independent research. Like, wow, so you have this period in early life where all these things are happening. The epigenome is in complete flux. DNA methylation goes from very low to very high. The polycomb landscape also undergoes this dramatic transformation. And there's this potential for things that happen very early to be remembered throughout life that could have consequences on physiological processes that, are postnatal. I think that, that's very interesting.
And this memory, comes this from the PRC2? Or what is the mechanism?
Well, so the model is, I think it's not completely fleshed out, but like what our working model, which has been our working model for years, is that when the denamethylation at this particular gene ZDBF2, I don't want to speak too universally, but at this one gene PRC2, when there seems to be denamethylation there, it seems to protect from these polycomb repressive complex 2 and maybe polycomb repressive complex 1 as well, from coming in and making this big polycomb domain, which has really stable silencing. So it's kind of a protective measure, I would say. And then if you remove that early embryonic denamethylation, the polycomb domain gets established, and then it can never get overcome, even with the proper transcription factors later on in life. So they just you have this really kind of stable silencing. So yeah, the memory of silencing would then indeed be the polycomb, but the memory of protection would be the denamethylation.
Okay, I see. So Our next paper was about the CTBF2 gene and how it's regulated, right? I mean, now everybody is talking about enhancers as well, but this was more about the promoter usage and which promoter was used then at this gene. So this sounds like a complex mechanism, and maybe also like can you maybe shed some light on this?
Yeah, I remember. So, we had this paper that was published in eLife, and I'm very proud of it. It hasn't been cited very much, I think, because it is quite complicated. And I remember that there's this comment on Twitter from this guy, Anders Hansen, who's a really brilliant a biologist studying 3D genome organization. And I remember his comment about my paper on Twitter was like, "Wow, this is complicated." I was like, "Oh wow!" So if this super brilliant guy thinks it's complicated, this is going to be tough to break through. But yeah, indeed, what we it's great, and this is an audio medium, so it's going to be it's going to be even harder, even more of a challenge to explain.
Yeah, I will link the paper in the show notes. So maybe
maybe we'll get more citations. Yeah, but essentially, there's for this ZDBF2 gene, there's actually two promoters. There's a promoter that is expressed in the naive pluripotent state, and then there's a promoter that's expressed in the primed pluripotent state. So there's a promoter switch that's happening during differentiation, or during this transition
on the same side, right?
Yeah, exactly. Yeah, and the one promoter actually the elongation goes through the other promoter, and we think that long isoform is actually required for setting the epigenetic state of the canonical of the shorter isoform. And anyway, in between these two promoters are these four enhancers. So what we showed was that there's three enhancers that are active in the naive state, and they're regulating the long isoform. And then as you have this epigenetic switch, the fourth enhancer gets turned on, and then the long isoform gets turned off, and those four enhancers then come over to the canonical isoform. So you have this kind of enhancer switch and promoter switch that's happening kind of simultaneously. And of course, this is all regulated in 3D space via action of CTCF and presumably cohesin, which is something that we showed actually during my most recent paper in my current lab.
Yeah, you're spoiling already my next question.
Because, sorry. Yeah, spoiler alert.
So this was basically then the story that also started your own, not started, but was the foundation of your work that you're doing in your own lab, focusing on, or at least the next steps were focusing on CTCF, 3D genome, and how this all acts together with DNA methylation, right?
Yeah, exactly. And I mean, I'm hesitant to say our lab really focuses on CTCF and 3D genome organization. It's really, it's really not the heart of the lab. But I think because we published that paper in a review, I now seem to be getting asked to review a lot of papers on the subject. So maybe it's kind of become that way. But what we do know, from work published, 2 decades ago, is that in certain contexts, most famously at imprinted genes, these are these class of genes that are expressed in a parent allele-specific manner, it seems that there is DNA methylation sensitivity, I should say CTCF is sensitive when its binding site is DNA methylated. And given that there's this dramatic gain of DNA methylation during early embryonic development, we thought, if we go from basically 0% methylation to 80% methylation, maybe we're going to be screwing up a lot of CTCF binding sites. And if we screw up a lot of CTCF binding sites, maybe that actually has impact on, gene regulatory programs. So, we had this really nice system that we wanted to, exploit. And, we found, and I should say, there are other people who did really nice work in this area, like Dirk Schubler, John Stam, I can't pronounce his last name.
Yeah.
That's a really long Greek name. Yeah. So it wasn't like these completely novel out of the blue questions, but we did have a nice system that we wanted to, use to interrogate it. And what we found was, about 1% of CTCF binding sites appear to be DNA methylation sensitive, which, is kind of glass half full, glass half empty. So 1% is not that many, but there's a lot of CTCF binding sites in the genome. So we're talking all of a sudden about close to 1,000 CTCF binding sites. You could imagine that could be impacting the regulation of a lot of genes. So the next step we wanted to do was see, okay, what's the impact on really 3D genome organization? And, of course, there's dozens of techniques you can do to explore this. We chose one called high chip. So it's basically like a high C, except you were enriching for chromatin binding factors or, histone modifications. So we did this for 2. We did it for CTCF, and we also did it for H3K27 acetylation, which is a mark of active promoters and enhancers. And the idea is in this way, we could look at how DNA methylation is impacting the CTCF landscape, how it's impacting the CTCF-mediated 3D genome organization, and then next step, okay, well, if we screw up CTCF, how's that impacting enhancer-promoter interactions? That was kind of the idea. Long story short, we didn't see that much. We didn't see that much. And I think what was kind of ironic was the gene that jumped out, number one hit, most significant differential loop that we found was that the gene I spent my whole postdoc studying, which was ZDBF2. So, and it was kind of a joke. The postdocs, Julian and Anna, who were working on this project, like, oh, they were kind of slapping their forehead, like, oh, I can't believe it's ZDBF2. And I was really happy. I was like, oh wow, it's ZDBF2. Like, I know how this gene is regulated. And I think this gene that I spent so much time studying is very clearly very responsive to the system that we're using. But that was really cool because that really tied that enhancer paper now with this CTCF paper. It was actually a really nice marriage because now we saw exactly how it was happening. This enhancer switch from these 3 enhancers going to one promoter to the 4 enhancers going to the other promoter that was entirely due to this denamethylation sensitive CTCF mediated looping. So it was really cool. It was really cool, and very satisfying.
I mean, the irony or the interesting thing of all those CTCF studies, the 3D genome studies, is that it's right in your face, right? You can visualize it, you can see it in HiC, you can see it in IF, you can visualize chromosome territories, all those things. But in the end, if you manipulate it, the effects of manipulating, removing TUDs, removing CTCF, whatever you do, it seems to not do much, right?
And I think you have to be a little bit careful too, because a lot of these papers are done in embryonic stem cells, which do seem very resilient to all these mutations and all these chromatin factors. So embryonic stem cells, these are mouse embryonic stem cells, of course. They're great because they're really nice for genetic manipulation. They're really nice for generating mutations in chromatin factors. But you also have to remember they're not a really good model for epigenetics, because they've kind of this really dynamic epigenome. So it's kind of the two sides of it. So what we try to do in our lab, we're not we're not the only ones, but we can muck around in embryonic stem cells, but then you always have to differentiate and see, okay, what happens when you kind of commit to a lineage, and then you can see how this might have more of kind of a, I would say, cellular impact.
When you're committed and when what you want to do is like silence the enhancers that you don't need and use those that you need. And the same is true for promoters and genes. And then if that's set, then maybe it has a better effect. If you then activate enhancers that should be in a liver cell and you are looking at it in a kidney cell or something like that.
Yeah, that's exactly right.
Yeah.
That's it's epigenetics.
Exactly.
It's cell identity.
Just stating the obvious. So the last thing I wanted to talk about is a paper that came out last October, and we are recording this in December 2024. And there you came back to the things that you mentioned at the beginning, right? 5-HMC, PRC2, H3K27 trimethylation, and the landscape of all this. And now you did this in a mammalian system and not in the plants. So what did you find there?
Yeah. Well, yeah. So this was this was kind of building off on this idea of denamethylation mediated antagonism of polycomb. So they just to very briefly state, we know that the denamethylation during epigenetic reprogramming goes in the pre-implantation state goes from very low, then as you implant it goes to very high. Whereas the H3K27 trimethylation, which is the histone mark deposited by polycomb repressive complex 2, in mice at least shows the inverse. You have these broad polycomb domains that then become very restricted, largely to unmethylated CpG island promoters that are silent. So CpG for the listeners out there, CpG islands are these very CpG rich regions, stretches of DNA that are very strongly associated with the promoters. I think something like 2/3 or 3/4 of mammalian promoters of these CpG island sequences. So we know that CpG islands are absolutely, they're chock full of transcription factor binding sites, and they have this very special regulation. But what we wanted to see is like, okay, the rest of the genome where this denamethylation is kind of coming in, maybe at the expense of K27 trimethylation, maybe that could have some kind of a regulatory impact like we saw at ZDBF2. So, we used our embryonic stem cell system to go from these naive to exit the naive pluripotent state and then look for regions where denamethylation really seems to come in and supplant H3K27 trimethylation. And then the second part was just looking at RNA-seq data and see like, okay, well, when denamethylation comes in, does that correlate with the gene turning on? And then we can find, okay, well, maybe it wasn't just ZDBF2 that's regulated like this. Maybe we can find find many. And so the genomics was beautiful. This was really thanks to these two postdocs I mentioned, Anna and Julian, who did this really nice. They were very synergistic colleagues. One was doing all the wet lab work, the other was doing all the computational analysis, and they had made these beautiful maps of denamethylation, K27 trimethylation, gene expression, and they could really hone in on those genes that looked like they were regulated like this. And I think we ended up with, I forget the final number. You can look in the paper. I think it was like 100 something genes like this. And then, we hit a bottleneck because all of this is kind of correlative, right? We're just looking at genomics data. And, when you look in different mutants for like methylation mutants or in polycomb mutants, you can have all kinds of secondary confounding effects. So something that like was really at the heart of the lab when I started the lab was to really implement these epigenome editing tools into the lab. And I know you interviewed Jamie Hackett, who's really been pushing this in his lab as well. And so we thought, okay, well, let's use these denamethylation editing tools, and then we can find our favorite candidates, and just in a wild type background, we can play around with the denamethylation, and then we can see, okay, it's really the denamethylation that's causing the antagonism of K27 trimethylation. So this was very simple in my head. It took years to get working. This was ultimately a heroic work from Teresa Early, who's a PhD student in the lab, who really ended up making it work. And then for your listeners who are interested in epigenome editing, or denamethylation editing, I should say, this is after years of us struggling and crying and frustrations. What works better than putting on denamethylation is taking off denamethylation. That's what really saved the project. So what we did was we differentiated from low to high methylation, and then we removed the denamethylation using these catalytic domain of these TET enzymes. And that was miraculously robust. This was just fantastic data. It was like too good to be true and highly specific. And then at our favorite candidates, we saw when we take out the denamethylation, K27 trimethylation remains enriched only at these regions, and that was associated with silencing. So this was, it was huge amount of gratification, because it was so much work trying to get it to work, and then when it worked, it just like it couldn't have worked more beautifully. So that was really, really exciting to publish.
Yeah, I can imagine that. So we are already coming to the final part of this episode. So imagine you are due to submitting a grant proposal tomorrow. What would you have written into that?
It's actually, I can relate to that because that's what we do. Yeah. So anyway.
So what would you have written into that?
Yeah, well, so there's a few aspects that I want to explore from the stuff that we already talked about. I don't want to give away like my whole research project, but there's a lot of aspects of this denamethylation polycomb antagonism that I think we can really kind of pull the thread on that and find mechanistically what's happening. We have some ideas, but we don't really know. That's something that's really kind of I'm very interested in. The other aspect of that is that ZDBF2 was so amazing because in the epi mutant, not the mutant of ZDBF2, but the epi mutant of ZDBF2, we had this postnatal effect. So, something that we were really trying to do now, and we have some, preliminary data, is to introduce our epigenome editing tools in vivo, change around the denamethylation, and see if we can see some impact that might not actually have an effect on the embryo or, certainly not the pre-implantation embryo, but maybe we could see kind of either more a fetal or postnatal effect. So, I think that's like a really exciting avenue to pursue. And then, two other things I didn't talk about at all, but, we're really interested in enhancer regulation. I think enhancers are, of course, this is not my idea that denamethylation enhancers is interesting. The enhancers I kind of stand out because they're one of the few elements in the genome that seem to be dynamically regulated at the level of denamethylation. Most of the genome is either methylated or it's not, whereas at enhancers, there seems to be this really push and pull between transcription factors and denamethylation. And it remains an open question, is denamethylation doing much or anything at these enhancers? Denamethylation could just be essentially just a readout of a silent enhancer. And we have some nice technology that we're implementing now that hopefully I'm saying it now, so maybe if I say it, it's going to happen. Hopefully we'll have a preprint out in 2025 on this side. And then the last thing, I mentioned earlier, my kind of my most deepest part of my heart is still interested in evolution and looking at, trying to answer some kind of evolutionary questions, looking across mammals and seeing, where DNA methylation might be doing something in one species of mammals, but not another. That's something that we're just starting to dip our toes into a little bit. So, that's right now. I can't speak too much about it because there's, we don't even have much preliminary data to talk about. But no, it's certainly where my passion is steering the lab.
Yeah, you already answered my question if you missed something important, but is there something else that you would like to add?
Wow, no, I kind of hit it. I would say that the starting the lab, kind of the transition from starting a lab to now the next step of the lab. That is kind of, I think you caught me at kind of an interesting time because we got our first papers out, and now we're kind of at this hub where we have to kind of make these decisions about where the lab goes. And it's kind of very exciting, and it's thrilling and also harrowing because, as like there's not, at some places, I guess you've got really nice institutional funding, but, most of us in academic research are kind of grant to grant. So it's kind of this, it's kind of, you kind of caught me at a kind of a very interesting time in the trajectory of the lab. But I'm very confident, I'm very excited. And I think, I'm very optimistic about how things are going to unfold.
So I guess it's the time where the first PhD students are finishing, exactly. And then you are studying new ones. Are you looking to grow your lab, or what is the best size for you?
Yeah, I was in a PhD lab that was very postdoc heavy and very PhD student light. But in the European system, or at least the French system, it's very difficult to have a lab like that. The resources to have that many postdocs is almost impossible, except for very select few. And on the other hand, I don't want. Well, there's also French rules in place too, but I don't want to have too many PhD students either, because I think PhD students deserve a lot of my time and mentorship. And if I had 7 PhD students, I'd be spread too thin. And hats off to people who can handle that. So I think for me, kind of, I'm at this stasis right now that I'm quite happy with, which is about 2 PhD students, about 2 postdocs, maybe 3 postdocs, and then we have a permanent researcher in the lab, Priscilla, who's has a French government position, but she's in our lab. And then we have a fantastic lab manager engineer, Margarita. So it's right now I kind of like this 7-8 person with master students coming in and coming out. I think to me that, at least for me, like with my personal style, I think I seem to be able to manage that fairly well. Wow. So you can ask them, but I think for me at least it feels pretty good.
So to finish, what is your personal definition of epigenetics?
That's what I always ask students. Nobody ever asked me. Yeah.
There you go.
It's a great question. And, of course, it feels like at the bar after a couple of beers is that type of stuff you can talk about for hours. What is epigenetics? When I talk to students in the lab, what we talk about is epigenetics in our lab, which I think is important, is a change at chromatin that has an effect on, a gene regulatory state. And that change can be inherited mitotically, from parent cell to daughter cell. That said, I always tell everybody, there's no epigenetics police. There's nobody who's going to arrest you for misusing the word epigenetics. For my parents, for example, who are not scientists at all, epigenetics to them is, there was a famine in Holland, and 2 generations there's there's more like diabetes because of something that happened to the grandparents. That's fine. Sure, that's epigenetics too. But I think as we're a real molecular biology lab, we're working almost entirely in cell culture. For us, this is, and we're also studying chromatin. So for us, this is really kind of our lab's definition of epigenetics. But as I said, do you have another definiation?
Yeah, definitely. I'm collecting those, and maybe we will publish them someday.
Yeah, you'll have 1,200. Yeah, no.
Yeah, maybe right now it's about 20. So.
No, I remember I was at a Gordon conference. This was years ago during my postdoc, and there's this guy, who's invited, who's talking about prions. I was like, prions? Like, well, that's not epigenetics. And you hear the talk, and you're like, well, why not? It's not genetic; it's inherited. So.
And it's having an effect on transcription. So
exactly. Yeah.
So there is no epigenetics police, but we have a nucleosome police. But maybe there will be
true.
a plus.
That's true. You have to always watch out for Karen Luger. Yeah. No, she's pretty scary. Yeah. No, I'm joking. She's lovely.
So thank you, Maxim, for your time and for being on the show.
That was a pleasure. Thanks for the invitation.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.